Okay, I forgot again. But I got a bad example. So if you then want to see, do these two classifications somewhat correspond to each other? Yes, no, and can I learn something from that correspondence? Then you need to do a partition mapping. So how does that start? You start with a contingency table. So basically, you draw up a table where you say, okay, I have 4 islets that have class A and class 1, 5, B and 1, etc. So you can either put that as a contingency table, or you put it more visually, where you say, okay, that group 1 has 9, and they're split up 5 and 4. Those 7 are C's, 3 also has 1 C, and 6 D's. And then you can try to start finding some mapping, and that is really a result partition that summarizes the input of those two. So you could say, okay, A and B are always type 1, C is always type 2, and D is always type 3. But then again, you have an error. So you could say, okay, that previous map was too strict, so I use another set of rules, or another set of maps where you say, okay, A and B correspond to 1, and then 2 and 3 are C and D. But then you see, you miss an observation there, so you can't really that call. So either you have rules, so you always have to make a trade-off. That's what biology is about. So when rules are too strict or too general, you can get either violations, so you have non-empty cells in your contingency table that are violations, so that are not covered in any rule, or you have missing entries, so an empty cell in that contingency table, so something that you did not observe. So as an example, I took the data from the Miro VNTR Plus website. So that database contains, I think that should be switched, 186, I guess, VNTR types, and about 90 different polygon types. So what did I do? I just compared the polygon patterns with the VNTR types. And what was the result? And that was quite surprising, because we didn't do that, it was just a test, that each polygon pattern corresponds with some VNTRs, and there were no violations in that whole set. So what does that tell you is that from your MLVA, from your VNTR, Miro VNTR type, you can predict basically this polygon pattern. So of course it's only on 180 samples, but still. So for example, Miro VNTR type 1.1 corresponds to this polygon pattern, etc. And then you could also put the more geographical names to your polygon types and learn some more of that. So that was the result. So they matched exactly without any single violation. And since Miro VNTR was the finder of the two techniques, basically that means that you can predict this polygon type from your Miro VNTR. So this is not a call to say you don't have to do any polygon analysis anymore, but it's just a way of showing what you can learn from data. So, to summarize, software is very useful, but it's not magic. It will not solve all your problems. So the principle is there are garbage in, garbage out. If you have a bad gel, if you have a bad sequence, it cannot solve or improve quality that's not there. Another thing where you have to be very careful with is that hierarchical clustering will always result in clusters. Even if you cluster random data, you will still get clusters. So always back up your interpretation with other information, epidemiological information, other molecular information, before you actually draw conclusions. And also try to understand your questions, your molecular, your epidemiological questions, and also know the limits of the algorithms and the coefficients that you're using. For example, know that for UPGMA there can be many different solutions. And lastly, software, it's just a tool. So it's really up to you as a scientist to make that final call and to draw the correct conclusions or make the correct interpretations. And I would like to thank you, and if there are questions, I'd be happy to answer them. So the initial idea was